All right, thank you, Chrissy. We have a few minutes for questions, and we do have a few questions that have come in through the Q&A. I encourage everyone to type in any additional questions you might have into that box, and we'll try to get to them if we have time. So the first question, when the phenotype is not well defined, like atypical presentations or undiagnosed diseases, how can we filter according to phenotype?
Yeah, it's a tough question to address. And it's something that we face very often in the clinical setting. Often we're looking at patients that don't really have a clearly diagnosed disease. And so the approach that I usually take is to try and break down the phenotypes as much as I can, and basically assemble a kind of mishmash gene list of genes that are associated with any of the phenotypes the patient has that are known already, and put those together as best I can. There are some interesting programs out there. One of them is called Phenomizer, where you can actually input all of the different phenotypes your patient has, and it tries to predict what the diagnosis might be based on that, and also will output some genes that are related based on those predictions. So we do use those as well, with the caveat that we never rely solely on those types of tools. They're just kind of a supplemental way of pulling out genes that might be interesting to your patient. But beyond that, that's why we also take a genotype-centric approach when we do these analyses, so that we're looking at the genes that we think might be related, but we're also just looking at any suspicious variation to make sure that we're finding any diagnoses that we might not have expected for that patient.
All right. Could you touch on what depth of coverage is sufficient for detecting a small proportion of heterogeneous genetic variation, say in a family-based whole exome sequencing study?
Yeah, so we usually aim for about 15x coverage to be able to reliably call heterozygous calls. We've gone down to 8x for that. But if you're looking at more mosaic type variation, so variation that has happened in somatic tissue, that is when you actually need a lot more coverage, so going up to 100x or more, to be able to reliably detect those kind of calls.
All right, and the tools and filtering criteria that you mentioned, are those limited to rare diseases or can they be generalized?
So they're definitely targeted towards rare disease, but they can be generalized. So you would have to make adjustments, for example, for your frequency cutoffs if you're looking at a more common disease. It does also get harder when you're doing gene list type analysis for common disease, because there's generally not very well established genes, or if there is a gene list, it's just so many genes that you might as well be looking at the whole exome anyway.
All right, here's another question that's come in. For extremely rare diseases, is statistical significance equally important with regards to sample size, or the consequence of is the consequence of the variant more key? And if statistics are important, what do you do with pathogenic variants that are not statistically significant with regards to sample size during reporting?
So it's a difficult question. When you have really extremely rare diseases, you're not going to be able to necessarily take a GWAS type approach, and it really is more of an N of 1 approach. So when we have a candidate variant from diseases like that, we really take a more targeted approach just to that variant. Instead of doing, statistics in a population, we'd be looking at the variant, trying to prove it has a functional effect on the protein, for example, trying to find other patients if we can, which is often difficult, and going down that more functional route, maybe an animal model if we can.
All right, and final question, considering that you check the frequency cutoff in Nomad for variant filtration, what happens if the variant is not captured in Nomad?
So if we're asking if the variant is absent from Nomad, that's actually something that we would capture in our cutoff. So that would be a very good thing in terms of, or at least it would be supportive that the variant might be pathogenic if it was absent in Nomad. I'm not sure if that was what the question was.
All right, and we do have a few other questions coming in. We do need to move on to our next lecture, but Chrissy will be able to type answers to your questions back to you in the Q&A while the next lecture is playing.